Hey class, so today we're looking at the brain, and this will be a four-part lecture. Now, you can start with just some wild diagram of the brain with lots of things labeled. This diagram here is a sagittally cut brain, but this is going to feel pretty overwhelming at first. You will learn all this stuff and their functions, but you don't want to tackle this by, I don't know, just picking at random or something that sounds interesting. You want a methodical approach to learning this many structures. So one approach would be just to take a nice big diagram and just go through one at a time, but it's going to help you a lot if you stick with the approach, and the same approach you'll see my lectures and textbooks will use, breaking down the brain into the major portions: the cerebrum, diencephalon, cerebellum, and brainstem. And in short, the spinal cord here attaches to the brainstem, and this is really some of your most primitive functions, right? Just kind of keeping your heart beating and your air coming in and out of your lungs. And as you advance to the more superior and even the back portion here, things will get more and more complicated. When you reach the very front tip here of the brain, that's where things like social niceties and culture and personality, planning for the future, things like that are found. So you want to definitely get used to this view. Realize there's a lot going on here, but also realize that you'll want a very methodical approach to tackle all this. And then, as you get more comfortable with saying the word diencephalon, for example, then you just get a little bit more detailed. You start to talk about the cerebrum being made of lobes. You can start to memorize the parts of the diencephalon, the parts of the brainstem, and so on. Look at different diagrams, watch some videos, look at pictures of models, and just slowly kind of add more and more detail to what you're learning. But before we get to any of that, let's talk about how the brain is protected and supported. Now, here's this short video clip that I wanted to show you of what a unfixed, right, non-preserved brain would look like.
But it's very, very soft. Notice, it's totally squishy. It's the consistency. It's much softer than most of the meat you would see in a market. So if I were to pinch this in either way, I could easily damage this with my thumb. In fact, neurosurgeons, when they are doing surgery, often just evacuate with a vacuum or suck out parts of the brain. This string here has been placed around the basilar artery so it can be suspended in a bucket of formaldehyde in order to denature the protein and harden it. If we didn't, it would sit on the bottom of the bucket and the brain would become deformed, as you would see here, so that it would be compressed in this direction just from the weight of the brain. So that points out one of the purposes of the cerebral spinal fluid is to float the brain inside the calvarium and act as a cushion.
Okay, so we will talk a lot about CSF today and as one of the mechanisms by which we protect the brain. But that's a great video to show how smushy and gushy that brain is. And you can watch plenty more. If you Google this term, you'll find a whole website, which is you'll be able to see all of these videos about the brain and spinal cord. They're also on YouTube, but this website has them more organized. So,
base here at the base of the brain.
Let's move on. We've learned about the bones of the cranium already, which protect the brain. So let's talk now about the blood-brain barrier, then the meninges, and then CSF. The blood-brain barrier, or BBB, is this required because the neural tissue in your brain and spinal cord and nerves, they have a lot of blood being supplied to them, but you really want nervous tissue to have a very specific, well-balanced environment. In other words, our capillaries are leaky. That's how we deliver oxygen and nutrients to all the tissues of our body. We just kind of make the capillaries pretty leaky. A bunch of stuff gushes out, and that includes lots of good stuff. But also it can include some rubbish that we don't want to necessarily reach a sensitive area like the brain. So there are both modifications to the capillaries, right? And there are astrocytes. These are two ways that the blood-brain barrier is tighter, more tightly controlled than other places in your body. And we could talk about that a little more. So When I say that the capillary cells are modified, I mean that instead of having cells kind of like this, where nutrients and things can flow out pretty freely, the cells are much more tightly connected. Now I'm going to jump down here. That does mean that in some places you might not be able to just rely on kind of things, the good stuff to just leak out into the brain when needed. You might need some pumps in place to kind of help move things against their concentration gradient. So it's a more kind of active process. And in addition to that modification to the capillaries, these astrocytes also cover the capillaries and the neurons. If we go back, we can see here's an astrocyte covering the capillary, so another layer of modification to the blood vessels and covering portions of the neuron. So astrocytes doing a lot of work to regulate this environment. And it's not perfect. Alcohol can get through, caffeine can get through, for better or worse, stuff like that can make it into your nervous tissue. But also, the hypothalamus and pineal gland are in the brain. And so they're hormone secreting structures. So those hormones need to get into the blood. That's what hormones do. So they actually have to have more porous areas in those areas to allow those hormones to escape the nervous tissue and get into the blood. So there's lots more that can be said about that. It's a pretty interesting concept. But the meninges are even more interesting. The meninges are the three mothers, meaning that there are three layers of tissue around the brain to help protect it. Now, this isn't actually a video clip, but again, you can watch this video and others by looking up this webpage. But I just snagged the screenshot. This is one of the three mothers, a really thick outer layer. So if you crack open the skull and pull the brain out, this is what you're going to find. You wouldn't really see the brain. It would be hidden underneath this thick connective tissue. If you remove that, there would be kind of this cobweb, ghostly, cloudy substance on top of the brain. And if you remove that, it would just look like a brain. But in fact, there's a tiny, thin layer of cells on top of that brain. So we're going to talk through the three mothers, but it's basically a super thick one, a kind of cloudy one, and one that's so tiny and thin you can't even see it. That thick connective tissue layer is called the dura mater. That's the first of the three mothers. It means durable mother or tough mother. It's actually made of two layers. The outer layer, the more superficial layer, is called the endosteal layer. So that's fused to the periosteum. It's fused to the skull bones, cranial bones. So ost, remember, is bone. Endo is within, so it's within the bone layer. Down here is the other layer. This layer is down towards the next of the meninges, right? We've got three meninges, right? So we're doing the most superficial. So we're going down towards the next of the meninges, which is here. So this is called the meningeal layer because it's connecting to the next meninx, which is singular for meninges. It's attached to the next of the three mothers. That leaves a space in between. That's called the dural sinus. So a sinus is a space, the space in the dura mater. And that is where blood is collected from lots of different blood vessels in the brain, and CSF is slowly and constantly being collected in the dural sinus. So that's a pretty important space. One more thing about this dura mater is that not only does it make a tough covering for the whole brain, it actually helps section off some chunks of brain matter. So these two terms here, the falx cerebri and the tentorium cerebelli, these are chunks of dura mater that are going to help separate parts of the brain. And we can see them on this next slide. Falx cerebri is the fold in the cerebellum. This fold of tissue goes right down the middle of the cerebrum, right? It splits the left and right cerebral hemispheres because they don't really talk everywhere. There's just a little middle part here where they can talk. So everywhere else they're separated by this thick band of tissue called the dura mater, but specifically the falx cerebri is the chunk of dura mater down the sagittal portion of the brain. Tentorium cerebelli, here it is pointing right here. right here, you can't see it, but there's a chunk of brain called the cerebellum here. This piece of connective tissue, the tentorium cerebelli, I think of it as like a tent over the cerebellum. I haven't looked up tentorium, but it's like a little tent on top of the cerebellum. Again, this is separating those pieces of brain. The cerebellum can't communicate with the brain through here, so it sends its communications through alternate pathways we'll learn about later. So, two modifications of the dura mater to provide some additional structural support more inside of the brain, not just around it. Next of the mothers is the spider mother, arachnoid mater. So this has these delicate web-like projections, processes, this meshwork. And what's really important to notice, this is the kind of thing that you'll get in Bio 5, but maybe not some more general A&P courses. The arachnoid mater is this membrane or layer. It's right here. I'm drawing it. I'm not touching the cobwebby part. So the actual membrane is right up against the dura mater. But then below the membrane are these little these mesh-like connective tissue strands. And this is forming what we call the subarachnoid space. So below the arachnoid mater is a space, subarachnoid space. This is where you've got a bunch of CSF flowing. When the video earlier talked about the brain kind of floating or being buoyant inside of CSF, this is the little bit of CSF that surrounds the whole brain and allows it to float in that CSF. So the subarachnoid space under the arachnoid mater is filled with CSF. CSF, as we'll talk about in a bit, is constantly being produced. Lots and lots more than you need is made every day. So it also has to be constantly drained away. So the CSF drains into these little spider-like pockets called arachnoid granulations. So it's where the subarachnoid space forms this little granule, this little bead or bubble. And right in this area, it's really leaky. So the CSF is constantly leaking out into this sinus. And then that will connect with the dural sinus. It'll mix with old used up blood, and it'll all go down the jugulars and back to the heart. In this picture here, you can see more of this cloudy spider-webby covering. That's all arachnoid mater. If you peel that off, it'll just look like a brain. But we do have one more covering left, the pia mater. If you are a musician and you know about piano or pianissimo means softly. So this is the soft mother. And this is attached right on the surface of the brain, following every little gyrus or sulcus, the grooves and ridges of the brain, right on top of them. So it's giving some protection, and it's helping anchor those blood vessels down. If we go back for a moment, we'll see here's pia mater right on top of the brain. And the blood vessels are kind of sandwiched in between the pia mater and the arachnoid mater. So they help capture those blood vessels. And then we have this cool picture here that I blew up on the next slide so we could really take a look and see if you did have a cadaver dissection that you were working on and you cut through the skin of the scalp carefully, right? It would be tricky to do this so well. You'd want to only cut through the skin of the scalp, and then you'd find the epicranial aponeurosis that the frontalis and occipitalis muscles are pulling on. But if you cut through that, then you would find the periosteum, the connective tissue layer on top of the bone. Then you hit the bone. In this case, you've got the parietal bones coming together on the top of the skull. Underneath the bone is the dura mater, or we know more specifically, first is the endosteal layer of dura mater, then you have the sinus, and then you have the meningeal layer of the dura mater. But if you pull through that nice thick connective tissue layer, then you get to the arachnoid mater. You can see it's kind of reddish here. That may be due to the fact that those blood vessels, remember, are in the subarachnoid space. There's that arachnoid mater. And then if you peel that off, you see what just looks like brain, but we have that super thin pia mater, this invisibly So it's such a thin layer, it's just invisible to our eyes. And that's following every little one of these bumps and grooves, these gyri and sulci. So that brings us to CSF, the last of the ways that we're going to protect and support the brain. The CSF is made by ependymal cells, which we talked about previously, but they specifically come from this structure called the choroid plexuses. Choroid means basically blood, and a plexus is a pleat, which I had to look up. It's like a sewing or fabric kind of thing. I tend to think of it as more like a braid, because when you see plexuses in the body, like those nerve plexuses we talked about recently, it's more like an intertwining of things. So I call it the blood braid, which is not totally accurate, but good enough. That's what's making CSF. Basically, the reason they're called blood braids is they're these modified blood vessels. You know, if you're going to make CSF, which is this clear liquid full of all the best yummies for the neurons, you need to make it out of some fluid that has lots of good yummies, lots of good nutrients and oxygen and so on. So you're basically just, you're allowing only the best stuff to leak from the blood in these choroid plexuses, and that's what becomes CSF. It's going to be clear. You're not letting out entire blood cells, red blood cells or white blood cells. You're just letting out some of those best nutrients. And your body has about 150 mils, but you produce more than 3 times that amount every day. So that means you're constantly making new CSF. You need to constantly allow it to leak away through those arachnoid granulations we saw earlier. So where can we actually find CSF? So we have basically these 4 places. It circulates around the brain in the subarachnoid space. But it's also in these chambers. You have these empty holes in your brain that you can kind of see a little bit here. They're called ventricles. Just like your heart has empty chambers filled with a liquid called ventricles, your brain has ventricles too. So you got it circulating around the brain in the ventricles. It drips down the central canal of the spinal cord, as we mentioned before, and the spinal cord has meninges, spinal meninges. They're just like the meninges we've been talking about, which means they have a subarachnoid space filled with CSF. So 1, 2, 3, 4 places you can find CSF. And then, of course, the CSF drains away through those arachnoid granulations we mentioned before. The thing about the CSF is, well, it's kind of complicated when you think about learning about it in lab, because you can have lab models like this. This is a structure that represents the hollow CSF filled spaces inside your brain. So you have to imagine if you took a brain and you injected it with like liquid latex, or I don't know, some other stuff that, you know, is liquid, and then as it cools, it hardens. And then you ripped open the brain, this is what would be left. So everywhere you see gray material here, that would be spaces in your brain. Everywhere you see like a little hole, a little space, that's represents solid. You know what I mean? So it's opposite. So that takes a little getting used to. But these are the ventricles and some other passageways in the brain that have CSF. We should mention other CSF functions. We said the brain is buoyant within CSF, can absorb shock, and helps transport nutrients and waste. So 3 pretty straightforward functions of CSF. So let's talk in more detail about what we're seeing here. Your brain has 2 lateral ventricles. So that's all this C-shaped thing with the little tail on the end. All of this has names, of course, but we're just going to say this is one of the lateral ventricles. Lateral is to the side. So you have one on one side, one on the other. We're going to see more pictures of this in a sec. 2 lateral ventricles. The next ventricle is right here in the middle, this round one with kind of a little beak down here. This whole area is called the third ventricle. So you've got one here, one here. They kind of drain down and meet up with each other to form the third ventricle. The fourth ventricle is way down here, and you have this skinny little tube called the cerebral aqueduct for drainage. These numbers I don't think correspond with this stuff, We've got two lateral ventricles. They drain into the third ventricle, which trickles down the cerebral aqueduct to reach the fourth ventricle. And from there, the CSF can drip down the central canal of the spinal cord. I got my alligator here because this third ventricle is kind of tricky to understand. It's laterally compressed, which is just a fancy way of saying it's like flattened but kind of tall, like the tail of an alligator. And so it's a very unusual shape compared to the other ventricles. So here's another kind of 3D version of this showing these big lateral ventricles with those tails. There's two of those. Then the CSF drains out of them through a space, which we can name the interventricular foramen, literally the hole in between the ventricles. The CSF arrives at the third ventricle, drips down the cerebral aqueduct to reach the fourth ventricle, and then travels down into the spinal cord. That's kind of your basic flow of CSF. You could do even a coronal cut of CSF and get another different view of this. And here you can see additional details like the gray matter on the outside of the brain and the white matter on the inside. You can see the septum pellucidum, a clearish wall separating some of these portions of the brain. But it's just to give you one more kind of look about how these ventricles kind of are separated and then come together in the brain. Last thing we'll mention here is just try to figure out what this all has to do with spinal taps, aka lumbar punctures, which is totally different from an epidural that a woman giving birth might receive for pain management. But they're kind of happening in the same place. And we referred back to this when we talked about the spinal cord. But if you want to either take a sample of CSF or inject some drugs into the CSF, the trick is to shove the needle into somebody's back without impaling them in the spinal cord. Now, one way you can do that is if you remember that the spinal cord ends around the first lumbar vertebra, then you can make sure your needle goes down maybe at L5 or something. So the spinal cord proper has ended. It's tapered away in this little structure called the conus medullaris, and then it's branched out into all these thin fibers called the cauda equina. So that's what we see here. Here's the cauda equina. Now there's less of a risk of this needle if it goes too far of impaling the spinal cord, because instead of a thick cord, there's just these fine little strands. So you need to have the patient kind of roll their back so you can get that needle in between the spinous processes of the vertebrae, through the dura mater, through the arachnoid mater, and into that subarachnoid space. That's tricky, and you can learn lots about this, but one way you'll be happy to know that anesthesiologists, for example, are learning to make sure that they don't accidentally jab one of the strands of your cauda equina is they put fruit in boxes and stick it with needles, and they just pretend it's you and your back. Studies have used, in this case, 4 different fruits that they put inside a box that would hide their identity. They labeled the fruits A, B, C, D. It turned out that it was an orange, banana, kiwi, and a melon. And they had the anesthesiologist, blinded, like couldn't see inside the box, take their needles and stick them through the fruit. And the goal, as the name of the title says, is to try to feel for the loss of resistance. I mean, if we go back, it's hard to push this needle through your skin and that tough dura mater that surrounds both your brain, but also in this case, your spinal cord. That's tough. You need to push hard, and all of a sudden, your needle will want to dive deep. It'll get through that hard part, and it'll want to just slide easy from then on. So you're trying to assess for that feel of loss of resistance. And it turns out that these different fruits give you a different feeling. And if you want to know the answer, I think it might be hidden. You have to look it up and see which of these fruits best replicated the feel of shoving a needle into someone's back. So next we'll talk about the cerebrum. Basically, we're just going to move through all the parts of the brain after this to learn about the different structures and functions that make the brain So smart. See you there.